welcome to Zoology 142 review. This is Kaono Hyaukai, and I'm going to be talking about cell-mediated immunity and its response in detecting a pathogen and the steps it takes to eliminate said pathogen. Cell-mediated immunity is kind of the other side of the coin as opposed to the other side of the adaptive immune response, which is antibody-mediated immunity. For the antibodies, they more target an obvious pathogen, whereas the cell-mediated immunity response eliminates infected host cells that have been, infected by viruses. And usually viruses will, infect the host cell and replicate within that cell body. So MHC1 complexes will take a piece of that virus or that antigen and present it on the outside of the cell, and that's where cytotoxic T cells or killer T cells will come and create that response. And from class we know that cytotoxic T cells are the one lymphocyte that directly attack pathogens. So let's just get right into it with this little overview right here. So first, the cytotoxic T cells will migrate to the site of an invasion, and they'll recognize from the MHC1 complex the antigen that's presented and bind to those target cells. And they're going to destroy these infected cells by secreting two substances. One is perforin, which will perforate the target cell membrane and create pores in the cell membrane of the host cell. And through these holes, granzyme will enter, and it'll degrade and release enzymes that will activate apoptosis. The next, this one is a more detailed, view and visual of what's happening in this process. So again, the T cell identifies the foreign antigen on the protein and binds. It releases perforin and granzyme molecules by exocytosis into the cell. The perforin will insert into the cell membrane, polymerize, and create pores in the membrane, through which granzymes will enter, activate its enzymes, and will trigger programmed cell death, which is apoptosis, and the cell will lyse and die. And after that, the T cell will detach and search for more prey, search for more things to kill, more infected cells. So yeah, that's pretty straightforward, I think. It's probably definitely going to be on the test, I would say. So what I suggest would be to memorize these little 5 steps, kind of get a base of that. So it recognizes through the protein, binds, releases perforin and granzyme. Remember those two. Perforin perforates, which means it makes holes. So I think that's a pretty good mnemonic to know. And then just know that it'll trigger cell death, and then it'll detach and know the T cells. So yeah, good luck on the exam, guys, and see you later. Aloha.